Go. We are good. So, Kaylin, we are setting this up to record, and you should be able to hear everybody, including some of the background noise here outside of the lower calf. Figured we'd come out here instead of being stuck inside of a classroom. So, first question, I put on this discussion series is: in what ways does universal common descent explain the variation in living forms? And the reason why I thought this was a good question is, we oftentimes think of the similarities among organisms as evidence of universal common descent. So we'll talk about similar structural forms or similar behaviors or similar functional identities as evidence for universal common descent, but then you have to still explain the alternative of that variation. These differences among forms, how do we fit that into this idea of universal common descent? So there's some of your ideas.
That's not one of my points I was writing out, that they split off into different organisms based on changing evolution.
Yeah,
time,
sure.
With some of them just being like little features, but then others being like big.
Right. So, yeah, and kind of thinking in terms of, organisms that have a more recent ancestor, that share a more recent ancestor, the differences between those should be more minor, that when compared to organisms where the most recent ancestor that they share is further back in time. And that would be true in the same way. The analogy I like to use often, it's not even really an analogy, it's just an illustration of this point. I am more closely related to my brother than I am to my cousin. Mm-hmm. And that's because the common ancestors that my brother and I share are our parents, whereas the common ancestor that my brother or that my cousin and I share are my grandparents. So my brother and I, the differences between my brother and I are more minor than the differences among my cousins and I, and that's because our shared ancestor is closer. And then if you go to third, fourth, fifth cousins, the distance you're going to the closest ancestor that y'all share is even further away. Therefore, the differences you would expect to be a little bit more obvious. Yeah, so that's kind of playing into your point, where over time you get some diversification, and some of those differences are minor, but some of those differences, are a little bit more pronounced.
So, the whole idea of evolution is that, like, with each generation, there are differences due to either mutations or other adaptations, and then the ones that are most suited to their environment are the ones that survive. And so, since there are varying environments and varying ecosystems, then different groups of the organisms will survive, like, according to their environments. So so each one is most adapted to their environment. Mm-hmm. And due to the varying environments, we would expect them to have varying characteristics.
Right. Yeah, and an important illustrating point for that is there are 5 main mechanisms of evolutionary change. There's natural selection, as, organisms, certain alleles are favored over others, survive better, and produce more offspring because of increased survival. Then there's sexual selection, where certain alleles produce more offspring, not because they're any more likely to survive or they survive longer, but because the features that they have are more attractive to mates. So those are 2 of them: natural selection, sexual selection. Then you have mutations. Mutations you mentioned as a way of adding new variants, or as a way of adding new individuals with a common variant. If you have a common mutation, you may not be adding a new form, but you may rather be adding a new individual with that form. So that's another mechanism. And then you have gene flow. And so this is a way of not necessarily introducing new forms, but introducing a new form to a place it hasn't been before, changing the ratio. So gene flow is emigration or immigration. So either bringing in new forms or losing forms, and it doesn't even need to be bringing in new forms, but it's just changing the ratios by bringing in new individuals. And then the last one is genetic drift, random changes in allele frequencies. And genetic drift is most often Most often is the result of some kind of a either a natural disaster or some kind of a founding event. Where a small portion of the population moves to a new area, or some random event kills individuals regardless of their form, regardless of how well adapted they are for their particular environment. So that's one good point to point out. It is absolutely, you're right, that you get changes at the population level that over time can result in kind of unique forms. So let's think about natural selection. Natural selection operating on height. Okay, so height is a polygenic trait, meaning that there are multiple genes that contribute to height. And polygenic traits are known for being shaped by environmental conditions. When you have multiple genes influencing the same character, environment plays a much stronger role in the phenotype than when you only have one gene contributing to a character. Does that make sense? Mm-hmm. And so with height, if you start to, for some reason, have an advantage of being tall, right? Where tall provides you some kind of survival advantage. So in humans, who knows what that would be, but in other organisms, it could be ability to exploit resources that you couldn't otherwise. And so there's some kind of survival advantage to being tall. And so over time, not only are more of your tall individuals going to leave more offspring, but over time, as you start to weed out short individuals, you could generate individuals that were taller than you've ever seen before, because you're weeding out the genetic material that produces short phenotypes. And as you start to wean that out and you get more purely tall genetic material, you can get taller forms than you've ever seen before. Okay, so that's another important point to make. And then one more thing I wanted to talk about with regards to your comment, Alyssa, is that there are basically two ways that environments change. One is through time, right? Environments change through time. As time passes, different factors contribute to changing environments, but environments also change through space. And so if an organism, or a population rather, not an organism, but a population is dealing with a changing environment, it could be that its environment is actually changing through time. So from one generation to the next, the environment is different. Or it could also be that the range of that species is expanding and starting to move into environments that are different through space and not necessarily different through time. Either way, you're dealing with changing environments. Changing environments have different pressures. Different pressures can produce different gene pools. Different gene pools produce different forms. Different forms could be described as separate species. Over time, you could explain the variation of life even with universal common descent. Make sense?
Yeah.
Kaylin, anything you want to add? Oh, that's right. Sorry, that wasn't very funny. All right. How does universal common descent help explain maladaptive forms or behaviors? Because again, with universal common descent, another thing you have to deal with is with the variation, right? It's very easy to say, okay, well, how does universal common descent explain similarities, right? How does universal common descent explain the fact that we have 70% sequence homology with, some carnivorous mammal, right? We would say, okay, well, there's 70% sequence homology because we share common ancestry. Okay, well, then we need to explain, how do we get that variation? And another thing we have to explain is, how do we have maladaptive forms? So how do we explain the fact that you have features that are not necessarily advantageous, that actually may be to the detriment of an organism? And so, for example, so there are portions, I want to use a marine form, but everything that comes to mind is a terrestrial form. So I'm going to use a terrestrial form, even though it's a marine biology class. So you have the Arctic hare. The Arctic hare has a winter coat and it has a summer coat, right? Summer coat is brown, winter coat is white. That is meant to correspond to the differences in temperature. There are parts of the Arctic hare's range where that color shift doesn't match what's actually happening in the environment. And so that would be an example that for that population, that is a maladaptive form. Okay? And so how do we explain that from universal common descent? Yeah.
The environment changed quicker than the adaptations could.
Sure. Yeah. So the environment shifted faster than the population could adapt to that shift. Yeah. And something that's always going to be true, the thing that is always true is a population is adapted for the environment of its parents. You know what I mean? Mm-hmm. The parents that were best suited for their environment produce the most offspring. So the offspring are adapted to the environment of their parents. And if the environment hasn't changed very much since their parents' generation, well, that's great. If the environment has changed since their parents' generation, it's not so great because they are adapted for an environment that may no longer exist. So yeah, that's absolutely one way to explain it, is that the environment changed more quickly than the organisms could keep up. Any other ideas?
Could one be that, say, a predator adapted faster than a prey? Like for example, like say with humans and insects, how some insects have adapted to our poisons. Well, we can create more potent poisons to kill them before they can adapt to the new poison.
Sure, and that would be part of the environment as well. So environments have biological and abiotic components. And so your competitors, your potential predators, your food source, all of that factors into that as well. So that we would just kind of put under the same category of the environment is shifting faster than we can actually adapt.
Okay.
By we, I mean, just a representative of that population. Yeah. Any other ideas? So yeah, that's absolutely a major explanation for it. Probably the one that most often is going to explain it. That whatever that feature is was advantageous under different conditions.
Would mutations be considered a component also?
Sure. That maybe natural selection hasn't had enough time to weed out some kind of a really common mutation, right? That the mutation is really common and leads to a certainly maladaptive form, and there just hasn't been enough time to get rid of that maladaptive form. Yeah.
It could be the maladaptive form isn't significant enough to really reduce the population.
Sure. Yeah, that it may not be ideal, right? But the thing is no population is perfectly adapted for its environment, right? This natural selection is an imperfect force, and so nothing's going to be, absolutely the best you can possibly have, for that particular niche of the organism. And the niche is just, there are two ways of thinking about the niche. You can think about the niche as the ecological role that the organism is playing, that the species is playing, or you can think about it as everything necessary for the organism to survive. So either way, both are true. Yeah. So that's another one. Yeah, I mean, natural selection just may not have had enough time to weed out a harmful mutation, or that it's just it's an imperfect force.
Well, like with people, like if we have some kind of mutation or something, it's not like they necessarily die right away, or they can never have kids.
Right. So the timing of the feature is a big issue. If the behavior or the form doesn't appear until after the individual reaches reproductive age, then it may not have as large of an effect. If it's not something that appears until later in life, then there's not much you can do. They may have already reproduced and passed on whatever that trait is prior to when the trait actually kicked in, when that phenotype came in. So there are two other major explanations for it, and these are again going to be important things to keep in mind. Because I'm recording this, I'll put the recording on YouTube. It'll be kind of weird because it's just going to be my computer screen. with than our voices. But you can listen through it and re-listen to this discussion. But yeah, there are two other big explanations. One is you have to keep in mind natural selection is not the only force. So Joseph talked about Jacob. Why did I call you Joseph? I'm so sorry.
Just sat next to each other. So.
Yeah, Jacob talked about mutations and how natural selection may not have had time to weed out that mutation, but you also have to realize that natural selection is not the only force. So it may be maladaptive in that it doesn't help the organism survive better, but it may be something that for some reason mates select for, right? And so like a peacock tail is a great example of this. The peacock tails, males with larger tails are more attractive to females, but it is maladaptive. I mean, it does not help them to survive better, and in fact inhibits their being able to move quickly in the event of predation. And then you also have gene flow and genetic drift, random genetic changes, and those random genetic changes are certainly not going to be adaptive forces. There's only one that's adaptive. The only force that's adaptive is natural selection. Some people want to make an argument that sexual selection is also adaptive, and I guess because fitness is a representation of how many offspring you produce, you could make an argument that sexual selection is also adaptive, but I like to think about it in terms of like just overall survivability. But I mean, you can make an argument that sexual selection is also adaptive. But so one, maybe two of those forces are adaptive. The other three are oftentimes not adaptive and are actually detrimental. So that's another big way to explain it. The last way that I can think of is sometimes maladaptive forms piggyback on something that's adaptive. That whatever that phenotype is controlled by a gene that's close to another gene that having the feature there is helps the organism survive better, and it's just that you have another gene that's close to it on the chromosome and it gets inherited as a unit. So like overall the organisms are more fit when they have whatever allele goes along with the important gene, and then you have this less important gene with a bad trait, but it just kind of is inherited as a unit with the good allele on a more important gene. So that's another one. We call that piggybacking.
Oh, I thought you meant like those.
Or coattails. Riding the coattails is another way to describe it genetically.
I thought you meant by piggybacking. Like there's these frogs that like even though they're small, what they do is they jump on. They would like piggyback off the bigger ones. And so when the big ones that are more attractive to the females, because they're stronger,
they sneak matings.
Right. Yeah. So that's
satellite males.
It's like little.
Well, yeah, I mean, there's another explanation as well. There are cheaters, right? Sometimes cheaters pass their thing on as well. So that's even another explanation. Is some people cheat the system, have figured out a way to cheat the system, and so they produce a lot of offspring, but their offspring are not like well adapted for the environment. They're not high quality individuals. They're just parents were good cheaters.
Would you consider, say, a human that has that was born with like an STD, like say hepatitis? Would that be considered a malform since they could potentially pass down to theirs?
Sure. Okay. Yeah, I mean, you can talk about a disease as a maladaptive form. Sure.
Okay.
Yeah. All right. Yeah, I mean, it's just there. I mean, you wouldn't worry about explaining it in the same way. You don't have to explain that because it's not a genetic form. It's not something that's, inherited. I mean, yes, it is inherited because they're oftentimes passed from mother to baby as it's being born, but they're not, it's not passed down in the same way.
Oh,
so,
okay.
Yeah. All right. Any other comments for that question? How does universal common descent explain maladaptive forms or behaviors? Okay. All right, so the next one is how do we model common descent? So what do we use to actually model this? Notice I didn't say universal common descent here because oftentimes that's really difficult to model because you've got, 1.8, 1.9 million described species, and if you try to model all of those back to a single ancestor plus all of the extinct species, it's just going to be insurmountable, But how do we model common descent? What do we use to model that?
We use the fork branches that break off from one organism to only two.
Yeah, cladograms. Cladograms, phylogenetic trees, they're the same thing, just different names for it. So cladograms or phylogenetic trees are absolutely a way in which we illustrate common descent. And so each branch represents a specific form, right? And so a really narrow branch, like a really small branch, may represent a single population of a species, right? Or more often it represents a single species or a single genus or a single family. And then there are bigger branches that represent still specific entities, but larger entities, like maybe an entire phylum or even an entire kingdom, right? And then each node or branching point, again, still represents a very specific entity. And you may not have any documented evidence of that entity ever existing, but it's still representing something very specific, the ancestor of the branches that split, the branches that combine at that node. Okay. Yeah, and so that's the way we tend to represent common descent. There are other ways of doing it. Prior to this thrust of cladistics, there were more rudimentary trees of life that weren't necessarily representing specific evolutionary events, which is what a cladogram or a phylogenetic tree is representing, is a specific evolutionary event, but we're instead representing just relationships, right? The way in which you kind of illustrating which groups are more related to each other than they are to other groups, but they were more rudimentary. And then there are other ways to do that as well. You can do it with like taxonomy and say, okay, well, if the organisms are in the same phylum, but they're not in the same class, that the ancestor they share is more distant than if they were in the same class. And if they're in the same class but not in the same order, then their ancestor is more distant than it would be if they were in the same order, right? So you can make inferences that way using more of a classical taxonomy as well, but we tend to represent the common descent using a phylogenetic tree or a cladogram.
But doesn't that time frame indicate like the speed at which they change?
Yeah, the length of the branch doesn't always represent the amount of time since they diversified, but it can. It doesn't always represent that. When it does, it makes that very clear, right? And you have that representation that the branch length is also a representation of time. But sometimes that is unfeasible in terms of having a diagram that's still easily read, that still has high levels of readability. Is that what you're asking?
Well, I was just saying that it shouldn't be, because like some species can be really different, but they have less time to evolve because their environments change so quickly. As opposed to something that maybe is like really distantly related, but they're really similar because their environments haven't changed.
Yeah, or because of just convergence for some other reason.
Right.
Right. So I mean, let's think of crocodilians and a lizard. Mm-hmm. Like so. Let's wait for this truck to pass by. It's a trash truck, in case you were wondering, Kaylin. Okay, so let's think about like an alligator and oh gosh, why can't I think of the name of the? I guess it's probably just called an alligator lizard. We have them around here, but let's just use a western fence lizard. It's probably one of the most common lizards we have around here. And an alligator, there are a lot of similarities in their forms, right? They look pretty similar. They're built in fairly similar ways. They're obviously going to have physiological differences. They have some significant anatomical differences, but their forms are pretty similar. But if you were to go back to the believed ancestor between lizards and crocodilians, you have to go all the way back to the ancestor of your first eureptilians, your first diapsids. I mean, you're talking about like 350, 400 million years ago to get to the relative of the supposed relative of western fence lizards and alligators, and yet they have very similar forms, right? And so, sometimes you just have, things that are believed to be convergence. Convergence is a fun thing to talk about in terms of how do you really explain convergence through universal common descent, where it seems like there are some forms that are just meant to happen. Like they just, there's certain designs that are just guaranteed to show up. And a lot of times it's really hard to explain why does the environment require this form, other than that there were specific designs that the creator had in mind, which I think is a better and clearer way of describing convergence. So, any other thoughts on how we represent universal common descent in cladograms, phylogenetic trees? Okay. So what characters are important for these models? When we're building these trees, what are some important characters to use to figure out, which groups are more closely related than others?
Rather than taxonomy, use specific features, but limit it to was one difference between the two.
I mean, if you wanted to get it down to that, to where there's only one difference between one branch and another, usually that wouldn't be the case, that you'd have multiple differences. But I'm just looking at, what are some important characters to use? So characters are just, like a specific, phenotypic suite, like eye color, hair color, various others. Yeah.
Something that's unique to that branch.
Okay.
So like feathers in the bird family, because they're only found in birds, nothing else. Well, I mean, other than crocodiles.
Theropods.
Yeah, other than them.
Yeah. Dromaeosaurs specifically. Yeah, and so I mean you could use feathers. Yeah, unique characteristics. So that absolutely that's one important type of character to use is our unique characters. You'd call those synapomorphies, derived characters, synapomorphies. And the whole reason why I'm asking these questions is because I want to kind of just get you into this idea of some of the advantages of universal common descent. Some of the advantages here. All right, so characters. Yeah, you can use unique characters that don't appear in other lineages. Sure. Like feathers in dromaeosaurs. Any other ideas? They don't have to be specific characters. They can just be, types of characters.
You go.
Characters that unify a certain group?
Sure. Yeah, no, that's good. So characters that are found in all of the individuals, right? Not obscure, characters that are only found in some of your forms. Good. And those make for good synapomorphies. And then if they are ancestral characters, you call those plesiomorphies. So synapomorphies are derived characters, things that are present only in that lineage and weren't present in the ancestor of that lineage. Plesiomorphies are still shared characters, but they're ancestral. So the example would be bone invertebrates, or let's say bone in fishes and bone in tetrapods, where the bone would have been probably in the original vertebrate ancestor, maybe not. Who knows? Because hagfish and lamprey don't actually have bone. So let's do bone in bony fishes and bone in tetrapods. That the idea there is that it is a plesiomorphy. It's a shared characteristic, but it's an ancestral characteristic. The ancestor had it, yeah.
Wait, hagfish are considered fish? They're considered like eels.
Well, eels are fish as well. Yeah. Hagfish are a different group. They're not in Osteichthyes, the bony fish. They're in Mixini. So they're in a separate category of fishes, but yeah, they're vertebrates and they're considered fish. They're not jawed fish; they're jawless fish, but yeah, they're fish.
Why are they considered fish and not like more closely related like snakes or snails?
Oh, that's an excellent question. Yeah, they are. They are believed to be more primitive, which and a better representation of the ancestral jawless fishes. The reason why they wouldn't be more closely related to snakes is then the idea would be that they have, a pretty recent terrestrial ancestor shared with snakes, which doesn't seem to be the case. But hagfish are just, they're an interesting group. I think most people now just put them as, a fairly derived and obscure jawless fish. So not a representation necessarily of the primitive jawless fish, but a jawless fish nonetheless. So now we have a fire truck. I hope they're not going to RCSM. Did they go past RCSM? Sorry, Kaylin, we're watching where the paramedics and the fire engine are going. Oh, they went around the corner. Okay. Yeah, they kept going down Placerita. Yeah, so that's kind of plesiomorphes, synapomorphes. So useful characters are in extinct forms. You would use a lot of morphological characters because that's what you have. Is you basically have form. You have form. You don't really have functions. You can't really use behavior or other things. You have forms. So you're going to use morphological characters. In living forms, a lot of the times we try to emphasize using molecular characters, using DNA sequence or protein primary structure, something along those lines, molecular characters. Because the idea there is that there's less bias, and again, going back to kind of this question, or not question, but this illustration, my brother and I are more closely related than my cousin and I genetically because we share a more recent ancestor, right? My brother and I share my parents, whereas my cousin and I, the most recent ancestor I have are my grandparents. So my cousin and I are more distant genetically than my brother and I are, right? And that makes sense. And then you go and say, well, why wouldn't you then think that the reason why we are more genetically similar to chimpanzees than we are to lions is because we have a more recent ancestor with chimpanzees than we do with lions. And the thing is, that's exactly what you would expect from the genetics, and that's why you would tend to emphasize using molecular characters.
Also, because some things look really different, but they're actually more genetically similar.
Sure.
And some things look really similar, but they're actually genetically really different.
Yes. Yeah, and that's where lizards and alligators are a great example. They look very similar, but genetically they are very, very different. Whereas crocodiles, crocodilians, alligators, and birds are more similar genetically to each other, but look less alike than crocodiles and lizards, at least on the surface. But crocodiles also have four-chambered hearts, like birds have four-chambered hearts. They are somewhat endothermic, like birds are endothermic. So.
Have feathers.
No, they don't, but that'd be awesome. That would be awesome, yeah. All right, so the next question is, what are some reasons to doubt universal common descent? What are some reasons to doubt it?
There's the big one, the polytomy.
Yeah, and that was something that you brought up about the video that we watched, which many of you said was not very good. And really, I didn't necessarily try to find the absolute best video on universal common descent, to be honest. I tried to find the one with the most views, so that you have an understanding of kind of what the most people have watched. And the thing is a lot of these are not going to be high quality, pieces. But yeah, I mean, anytime you have an unresolved polytomy in a phylogeny, that's an issue. So a polytomy is where instead of having a node split into two branches, the node splits into three or more branches. And that's an issue for several reasons. The biggest of which is that's not how we see microevolution and speciation work, right? The way we see microevolution and speciation work is you basically take one original population that over time becomes two sexually isolated, reproductively isolated populations. Yeah.
But that can happen in real life, like the finches that Darwin saw. Like, so the finch population, they spread all over the place.
They
like
did, but still
places.
split by split.
They don't split to like 3 places, and then those 3 ones kind of separately from each other.
Yeah, but then even when you go and you study them, and if you do a cladogram of those populations, they link to unresolved polytomies, to where 2 of those islands are more closely related than they are to the third island.
Okay.
So it's still, even though in time it may have been, the founding population went to 3 different islands. In reality, gene flow, because you have to realize it's not just about founding, right? That genetic drift, but there's also gene flow that's connecting them. 2 of those islands are going to have higher levels of gene flow than they do with a third. That's going to preserve some of those changes, and that's what's going to happen always. And so what's interesting is when you can see, no doubt, speciation generated the different forms that we have, right? With like an archipelago. You see that all the time with the Galapagos Islands. You see it in archipelagos in the Caribbean and archipelagos in the Mediterranean Sea and archipelagos all over the place. You can find examples where it's like, oh, this is clearly speciation happened to take one species and generate these different species that we have, which no doubt about it. Well, when you go and you build cladograms or phylogenetic trees to represent that, you don't get unresolved polytomies. You get it to where some of those populations are more closely related than others, and so you can actually even trace the events that happened or measure the amount of gene flow connecting them. And so when we look at a phylogenetic tree and we have an unresolved polytomy, when you say you have a phylogenetic tree of arthropods, right? So of all of your insects, crustaceans, myriapods, chelicerates, your spiders, your scorpions, all of those organisms, and you have an unresolved polytomy that basically you can't say what's more closely related to the insects and crustaceans, the spider or the millipedes and the centipedes, the myriapoda, and you have this unresolved polytomy where one branch splits into 3, it's evidence against universal common descent. It's evidence that the reason why we can't even model the ancestry is because that transition never took place. Right? Absolutely. It's great evidence against universal common descent. Any other reasons to doubt? Yeah.
When we see speciation, it's generally a narrowing down of the genome as opposed to widening out. And all of evolution is based off, you have a really primitive, or like a more primitive organism, well, not organism, what do you call it, population, and then those branch off into more complex ones, but we only see opposite. We see a more, not more complex, but more genetically diverse organism spreading out into different, less genetic.
Yeah, the most common type of speciation is done by what's called heterozygous fractionation. And so where you take an individual that had a great deal of variation, a lot of different alleles, and you start to get rid of some of those alleles, right? Like, narrow it down, make it more specific.
Like the finches. Like
yep. Yep, absolutely. And so that's an interesting thing. And so you have to have some kind of a mechanism by which you would increase the size of the genome, which there are proposed mechanisms for that as well. Gene duplication events, entire chromosome duplication events, which is interesting. You have some evidence for that in that, vertebrates have more master control genes than invertebrates do. And it's like, okay, well, if these master control genes, the genes that turn on whole suites of other genes for developmental purposes, if you double those, you get some kind of a duplication event, and now you have twice as many. Your original can go on functioning like they were before. Your new set can actually start to turn on different suites of genes and accomplish things you haven't seen before, right? And so we have the mechanism by which we can duplicate genetic material. So, but it's not, that tends to not happen during speciation events, right? Speciation events, you're right, tend to be more like heterozygous fractionation, where you take a lot of variation and you cut it off. And this is like with dog breeding. That's exactly what dog breeding is. It's heterozygous fractionation. You just start, weeding out some of that variation and start selecting exactly what you want, right? There's variation in size, and you want small individuals, so you get rid of all the large individuals, right? And you're eliminating some of that variation. Any other reasons to doubt common descent?
The plant tree, how first we have a cell that gains mitochondria, but then when it goes to plants, they immediately drop it.
Oh no! Plants maintain their mitochondria. Yeah.
What was it that we talked about that?
Well, maybe the endosymbiotic theory in terms of plants and their chloroplasts.
Yeah, it was that.
Yeah, and so you've got in plant cells and in a lot of photosynthetic organisms, it doesn't look like you have a good case for primary endosymbiosis, in which you had a eukaryotic cell phagocytize a photosynthetic, prokaryote and allow it to exist, but rather phagocytize a eukaryotic cell that had a photosynthetic cell inside of it, and so you got some kind of a secondary endosymbiotic event. And this is really difficult. Hang on, we have another trash truck driving by.
Might be the same one.
Probably is the same trash truck, just going the opposite way. That was from Alyssa, and she's likely correct. So now I got lost in my thoughts. What was I saying? Oh yeah. So it's an interesting explanation for the presence of mitochondria in the presence of chloroplasts, right? You phagocytize an alpha proteobacteria in terms of getting not the photosynthetic to get you your mitochondrial ancestor, or you phagocytize a cyanobacterium in order to get your chloroplast ancestor. It's a really, really interesting idea. It's an explanation for why do mitochondria and chloroplasts contain DNA. Why do they divide separately of the cell? The problem is you need more mechanisms. It leaves more unexplained than it explains. The fact that why are some of the genes for proteins that work in the mitochondrion actually in the nucleus? Why does the mitochondrion, why is the language of the mitochondrion, the genetic code, different than what you find in bacteria and in eukaryotes? It has kind of its own unique genetic code, where the genes that are transcribed in the mitochondrion that then get translated into protein have a unique genetic code, and we need some mechanisms. So it leaves a lot unexplained, and then when you start adding in like these evidence of primary and tertiary and even quaternary endosymbiosis, it just gets this really complex mess as trying to explain the origins. Is it a way, is it an explanation for it? Sure. And does it explain some of the reasons why chloroplasts have two membranes surrounding chloroplasts, and mitochondria have two membranes? That the original membrane is from the bacterium and the other membrane is from the phagocytic event, But the interesting thing is the membrane, the inner membrane of the mitochondrion does look structurally like the membrane of an alpha proteobacterium, but chemically it's very different, And so you got just some interesting questions that you end up having to answer. So yeah, I think it presents more questions than it actually answers. That's an issue. Another big evidence against universal common sense. Do you have some more reasons to doubt it, Alyssa? I'll give you a couple more.
You probably have some.
Oh, if you want to talk, yeah.
Just about the thing about the narrowing down though is the ways in which they explain genes being added are almost never to the benefit of the species. In fact, I don't think we've ever seen a pure benefit. Sometimes a partial benefit, such as what's it called? The blood thing, the sickle cell disease.
Right.
People use that, but it's not purely beneficial.
It isn't, and it's only under certain conditions where it provides any benefit, and it's only beneficial to be heterozygous, not to be homozygous for sickle cell disease. To have two improper forms of the hemoglobin. This is a good point.
I talked to this Italian scientist, molecular biologist one time for a long time, and I asked her can you give me just at least one example of one time that a mutation was useful to the species, and she couldn't give me a single one. And that's her job.
Yeah.
And if you're relying completely on something you've never seen, it's a bit of an issue.
It is an issue. Yep. No, you're right about that. Do you have some other reasons to doubt it?
I remember we had talked before about how there was pollen found in the Paleozoic layer when angiosperms had shown up in the Mesozoic.
Yeah, actually, so angiosperm pollen is an interesting one. Angiosperms don't show up in the fossil record until the Cretaceous. And angiosperm pollen basically shows up at the Cretaceous. Now, there is some pollen that is arguably angiosperm. That shows up earlier in the Mesozoic and maybe even in the end of the Paleozoic. But the problem is, pollen is probably one of the best examples of something that what you fossilize is going to look simpler than what it actually was like when it existed, right? So, through time, geological activity erases some of the unique features of material that gets fossilized. And if something like pollen, when you're looking at, your unique features are going to be, really small, intricate features, the ability to preserve some of those for a long time is difficult. And so your fossilized pollen is probably going to appear more primitive than what it actually appeared when it was made by the plant. So that's an interesting thing. And there's arguably some angiosperm pollen appearing earlier, but I think the whole angiosperm pollen issue is probably a better argument against a young Earth than an argument for it, because you really don't have undoubtedly angiosperm pollen until angiosperms start to show up in the fossil record, which is in the Cretaceous, the end of the Mesozoic. Whereas if angiosperms were there from the beginning, which is what the Bible describes in Adam being placed in the garden, and it talks about the fruiting plants. So if you've got angiosperms there from the beginning, you would expect to find angiosperm pollen even before they may actually show up in the fossil record, because angiosperm pollen is everywhere. I mean, we find it in marine sediments, we find it in sediments in Antarctica, we find it basically everywhere. I mean, angiosperm pollen gets everywhere. The wind blows it everywhere. And so the fact that it doesn't actually start showing up in the fossil record, undoubtedly, until you start finding angiosperm fossils, is a good argument that the angiosperms didn't exist until then, which is strange, right? And that I think is an argument against young Earth creation. Yeah, listen.
Okay. One of the main arguments for evolution is like saying that relying on the fact that whatever's happening now, the rate in which like things decay
Sure.
and things happen now, was the rate it's always been.
Yep, uniformitarianism.
Yeah, that was one of the main reasons for like universal common descent. Anyway, but in terms of fossils, most fossils are made in events that are not uniform.
Correct, are made in catastrophic event. Uniformed events do not bury an organism fast enough for it to be fossilized.
And so the fossil record shows not the uniform,
but a series of catastrophes.
Right.
Yeah. No,
which means that we only have, so we'll have a bunch during one catastrophe and like none when there's no catastrophes, which means you can't make a complete phylogenetic tree based on just random catastrophes.
Correct.
It's not going to be complete or reliable.
Yeah, you're right.
So the fossil record isn't as reliable as we like to make it seem.
Correct. Yeah. Yeah, so some other arguments against universal common descent: when you have a lot of variation appearing all at the same time, when you have a great deal of variation appearing all at the same time, that is an excellent argument against universal common descent, because it's just unexplained variation, and that's really tough. That's really tough to do. Some other arguments against universal common descent, probably the most important one is it's inconsistent with the biblical narrative of origins, universal common descent. What the Bible describes is God creating organisms after their kinds in separate events. And so universal common descent is inconsistent with the biblical narrative of origins. And that is probably the most important. And then you guys watched a couple of videos. I think it was maybe 25 minutes total of Michael Behe talking through irreducible complexity. And he talks about irreducible complexity being a good argument against universal common descent. And that there are some machines that are so sophisticated and so intricate that you cannot build them piecewise. They have to be built all at once or not at all. All at once or not at all. And so that's a big problem, and that's an excellent argument against universal common descent, is these irreducibly complex machines. Yeah.
The one thing that I have a little bit against that argument, though, is that evolutionists never describe things as evolving completely separate from each other. They don't say like, okay, the bones evolved and then the eye evolved. It's like, well, they evolved together. So it's like with one of the irreducible complex organisms I think people talk about a lot is like the giraffe. They said, okay, there's like a sponge in their brain that absorbs the blood flow, so that way when they drink, they don't explain the wrong.
Retain mirabile.
Right. So like, well, so you have a perfectly formed giraffe.
Yep.
And then it would blow his brains out, so it doesn't work. But it's not like evolutionists think that your brain evolves and then separately the sponge. It evolves together.
Sure.
So as it grows, the sponge grows a little bit bigger each time. So that's a similar thing with.
Yeah, I think the only issue, and you're right, it's not necessarily, nobody would argue. Like, let's say we use the flagellum as the machine that we're talking about. Well, I don't know if anybody's going to necessarily argue that the different components of that machine developed completely separate from one another. I think the challenge that you have is the fact that now, instead of developing something through microevolutionary mechanisms that we see at work, natural selection, sexual selection, genetic drift, gene flow, and mutations, now you have to start saying, okay, well, how do those forces produce a large enough change to generate this entire machine at the same time, right? So if we're not going to say, oh yeah, but we're not going to say that they evolved, like the different parts of the machines evolved separately of each other. But then the question is, okay, but by which mechanism did we generate this machine? You know what I mean? Like, what did that? What facilitated that? Because that is an issue, and that's something that you'd have to be able to explain. How does natural selection generate this fully functioning machine from nothing? I mean, it's not generating it from nothing, but from a non-fully functioning machine. And no pieces to make it. So you have to generate the pieces and assemble the machine at the same time. So.
I think it's more of an argument, I think, for how life originally came to be than how it narrowed it down.
Sure. Yeah. Sure. Yeah. And then what's called the edge of evolution. It's another idea from Michael Behe. I don't think he talked about it much in the videos that I had y'all watch, but he wrote a book called The Edge of Evolution, and basically the idea is here, we've been working with E. coli for what equates to millions of years in something with a longer generation time. E. coli, they have a generation time of 20 minutes, and so and we've been working with E. coli for just decades, and so it's equivalent to billions of years of time for something bigger, and we have been able to accomplish things. Right, there have been groups that have generated E. coli that can metabolize something that no naturally occurring E. coli can metabolize. That now they have E. coli that can eat a novel food source, right? And that is undeniably that's something new. Right? Mm-hmm. That's something new that was generated in this group. The issue is, in all of this work that we've done, in what equates to millions of years of time in something with a longer generation time, the only thing we've been able to do is generate E. coli that can eat something that E. coli couldn't eat before. Mm-hmm. But they're still undoubtedly E. coli. Mm-hmm. And E. coli have the same strain that they were to begin with. Mm-hmm. So we haven't generated a new organism, we've just generated an organism that can eat something new. Mm-hmm. That's interesting. So it seems to be that in all of these areas where we've done high levels of research on the evolution within this group, is that there's a very clear limit to what you can accomplish. Mm-hmm. There's a very clear edge of that evolution.
So would that more so be just changing its behavior from changing diet?
It's not just behaviorally different, it's physiologically different.
Oh.
I mean, so yeah, I mean, is it eating something that it couldn't eat before? Yes, but it's not really a behavioral difference, because they'll eat whatever's on the plate. It's a physiological difference. Like they can actually, they have the enzymes to process that material, and no naturally occurring strain of E. coli have those enzymes.
Oh, okay.
Then you have the question of, was this contamination? Did their plates get contaminated with something that did have the enzyme for processing that material, and then bacteria are notoriously good at transferring material horizontally. So is this an example of contamination rather than an example of evolutionary change? I don't know. But even if it is evolutionary change, we've accomplished developing a strain of E. coli or a group of E. coli that can eat something that E. coli can't naturally eat. But we still haven't generated anything new. It's still E. coli. So it seems to be that there's an edge to what we can accomplish. I mean, think about breeds of dogs. We have everything from a Chihuahua to a Scottish Hound. What do you call this? A Scottish Bear Hound, or whatever you call those massive dogs. And you're like, wow, it's a lot of variation. Yeah, but they're both still obviously dogs, right? You still look at them and you're like, yes, it's in the case of a Chihuahua, it's a sad dog, but it's still obviously a dog, With the Irish, man, I can't think of exactly what they're called. I think the Irish Bearhound, Irish Mountain Hound. Anyway, it's just this massive dog, and you look at it, you're like, oh, it's really impressive, but it's still obviously a dog, right? There's and we know exactly what we want, and yet it seems to be that we're still limited in what we can accomplish. We're limited by the variation we have to work with, and we're limited by the boundaries of just what's biomechanically possible. And you could argue that we need to shift it far enough away from its adaptive peak to accomplish something really crazy. But I think we've put organisms under the kind of stress that would produce that if it was possible, and it hasn't happened.
But I know a lot of evolutionists would just say like, oh, it just takes more time than that.
Sure.
So.
Yeah. You have other issues. Time is it's a benefit for sure, but it's also an issue in that natural selection, can't keep up with the deleterious mutations. And so you see, actually this year, John Sanford is coming to speak at our Creation Summit this year at the end of February, and he wrote a book called Genetic Entropy, and he actually came to Christ studying genetics and realizing that we know exactly what we want, and we try to get rid of everything we don't want, and yet we still can't get rid of some features fast enough to keep our strains from going extinct. So he's a plant geneticist, and he generated he holds hundreds if not thousands of patents on different plants, and he knew exactly what he wanted, knew exactly what he didn't want, and yet he couldn't keep up with the buildup of deleterious mutations that are what are called nearly neutral. So they're not devastating enough to weed out very quickly by natural selection. They just kind of accumulate to a point at which the entire gene pool just collapses, and whatever that organism is goes extinct. And he actually uses this as an argument for why you'll have an Ebola outbreak, and then all of a sudden it'll just go away. And it's not necessarily like we're great at holding quarantine, because obviously we're not. The last time we had an Ebola outbreak, I think you could see how poorly we kept quarantine. People were getting infected all the time. And he would argue that it's just, that particular strain of the virus just eventually, because of the accumulation of these nearly neutral harmful mutations, just gets to a point where it collapses and it goes extinct. And he would argue that that's the fate of everything. Every species on Earth and every form of life on Earth has the same fate, that natural selection, that if artificial selection can't keep up with the buildup of those deleterious mutations, natural selection certainly can't, because it doesn't have the foresight to know exactly what it doesn't want. So it's even less sensitive than artificial selection, and it's not going to be able to keep up with the buildup of those nearly neutral deleterious mutations, and that eventually everything's destined for extinction. And so time in that case becomes an issue, becomes a big issue, because it doesn't do a good enough job. We have basically too much time for the amount of deleterious mutations that we have in our genome.
Yeah.
So, yeah, you have some interesting issues also presented by time.
Yeah. Well, because I heard that they in general we think of it as like evolution. We think of it as like a tree branching off, where what we see is actually a tree upside down, which means within the uniformitarian idea that everything has the way it is now is basically the way it's always been, within reason, obviously. Then, in general, like if you look at species way back when, they were more genetically diverse, they're more like fit for their environment, and now maybe they're weaker and they're getting more extinct. So in general, we see a shrinking of the tree as opposed to branching out, which means that in general, over time we'd have less species than we have now.
Yes.
So it should be a shrinking, not a growing. So either it had to start with a bunch and then end up shrinking. That's what we see happen now.
Okay.
So you have to either violate the idea of the uniformitarian or the idea of like the branching. One of them has to be compromised.
Yeah.
They don't work together.
Yeah.
Because what I heard like a lot of evolutionists say is that the reason why it's okay to have like them growing as like the species and the adaptation level of the species is growing as opposed to shrinking is because the environment in which like life started or used to be was more like advantageous to evolution. Where now we're in an environment that's less like.
Sure. Yeah.
And so now we're dying out. Back then we were growing, but that means the idea of uniformitarianism like doesn't work. It's opposite. So you can't measure things the way it was if it was so different.
Yeah, unless you still are moving towards something in a uniformitarian way.
But
right.
It's going backwards, like slowly.
In terms of the number of species, but the actual physical environment could be changing in a very uniformitarian way. That it's progressively getting less favorable to change. You know what I mean? Like it started somewhere to where the early Earth was more favorable to change, and now it's progressively getting less favorable to change at a constant rate as it changes. Because I don't think you have to just assume, okay, because you have these physical entities, right? You have these laws, these mechanics that govern the world that have always been in place. That doesn't necessarily mean that individual ecosystems and even the biomes as a whole and even the biosphere as a whole, it doesn't necessarily mean that isn't dynamic and isn't in flux. And you see this. I mean, things tend to be cyclical, and so it doesn't mean just because like uniformitarianism doesn't necessarily mean that you can't have cyclical patterns in climate, right? Or you can't have cyclical patterns in ocean level or cyclical patterns in the level of lakes, like the Great Lakes. No, I mean, we even see that. We see this kind of cyclical nature to the physical properties that are present in the biosphere. It just means that the laws that govern the mechanisms, like the mechanisms at place, aren't changing, and the rates of decay or the rates of these chemical reactions that life depends on aren't changing. But you could still have this kind of cyclical nature of things, and so you can have even a cyclical nature to favorability to life, where there are certain times in which Earth's atmosphere and conditions on Earth are more favorable to life, and therefore you get kind of an explosion in the diversity of form. And then you cycle to a time that's less favorable to life, and so you get a regression. And then you could cycle back to a time where it's more favorable to life, and you get another explosion of the diversity, and you could cycle back to where it's less favorable.
The viewpoint that was brought up in my ecology class, where about how we, as God's creation...
Sorry, Kaylin, my computer died for a minute. I had to get it plugged in, so we lost the recording for a little bit.
About how we, as humans, are supposed to be stewards of the earth, and so it's given for us to be able to do what we want, such as build these structures that we've had, and anything of the sorts, but at the same time we have to be stewards and taking care of it.
Sure.
And so,
Yeah, we'll actually talk about that a lot in this class. How do we balance, how do we model our dominion after Christ, Because the best example we have of somebody having perfect dominion is in the way Christ has dominion over his church. And if we're to model having dominion over God's creation the way Christ has dominion over the church, it's modeled by sacrificial love, right? And that's not to say that we give our lives for a non-human part of God's creation, but it's, you know, we dedicate time and energy and resources towards thinking through how do we get God's creation to thrive the way God designed it to. All right, we'll talk about those specifically, so it's not so abstract. Where it's like, oh, like what does that even mean? Last question that we'll deal with. I know the last one says be ready to discuss the interesting marine creatures, but we're running out of time. We're already at 3:08, and we've had some good conversations. So how do we figure out which organisms do share ancestry? So this is from, we just talked about why should we doubt universal common descent. I think there are a lot of really legitimate reasons why we should doubt it. The main one is that it's inconsistent with a biblical narrative of origins. And so how do we know which species do share ancestry?
From a Christian perspective, we just focus on microevolution.
Okay, of what you could accomplish through microevolutionary changes. Yeah. Okay.
Whether or not they can reproduce.
Yeah, whether or not they can form viable offspring. Yeah, good. I like that. And really, we're just looking to where is it mechanically possible, not.
Does it happen in real life?
Right. Not does it actually happen, and really we're looking at is it mechanically possible at the cellular level, not is it mechanically possible at the organism level, because it's not even mechanically possible for a St. Bernard and a Chihuahua to reproduce at the organism level, and yet you can do it, right? You can do it in the labs. At the cellular level, it's mechanically possible. And so we're really looking at, at the cellular level, is it mechanically possible for hybridization? You're not even looking at, do the offspring that they produce, are they viable? Can they reproduce themselves? Because there are a lot of issues along that way. You're just looking at, is it mechanically possible? Yeah.
But it's mechanically possible. They made a spider that's like half plant, half spider.
Yeah, but not through normal fertilization. They did it through probably genetic engineering.
Right.
Yeah,
So
that's a different thing altogether. That's not normal mechanical, interaction of cells. That's genetic engineering. I mean,
Okay, so our definition is like, can they produce viable offspring via
mechanically at the cellular level.
Gotcha. Okay.
Yep. Yeah, I think that's a good characteristic. And if they can, then it's probably a good indication that they are part of the same original created form, the same created kind. And if they can't, then it's, I think, evidence against them being in the same created kind. I think there are other things you can look at. Do they share more features than they're different? Do they have more in common than they have not in common, if that makes sense? So when you think about humans and chimpanzees, sure, we have some similarities, but the question is, do we have more obvious differences than we have obvious similarities, right?
What do you mean by obvious? Because like on a genetic level, we're like 98% similar, but that's just because the genome is like similar for all animals.
Right, well, and that's only in the coding region, which is about 30,000 genes, which, is less than 1% of the total genome. So when you actually compare the entire genome of a human being to the genome of a chimpanzee, it's not anywhere close to 99% sequence homology. That's just in the coding regions, which when you're thinking about the genes, the mechanics, I mean, I don't think we should have issues with that amount of sequence homology. If it's a good design, it's a good design, right? The question is, if you're going to take the entire genome of a human being and compare it to the entire genome of a chimpanzee, which we can do. We have the human genome's been sequenced, the chimpanzee genome has been sequenced. You could do that, and I don't know what that level of homology is, but it's nowhere close to 99%.
Okay, because I heard we have like 90% similarity to a banana.
Oh, yeah, in terms of coding sequences, but bananas, I don't know how many genes bananas have, but yeah, I don't know what percentage of the genome you can even use in that comparison. I don't know. I don't know. And if you're just picking one gene at a time, I mean, if you do like cytochrome C, which is a gene, it's a mitochondrial gene that codes for cytochrome C oxidase, one of the machines in the oxidative phosphorylation. I mean, if you just compare that gene from one organism to another, or any organism that has aerobic respiration, by necessity, you're going to need some similarities in the way that that gene is structured, and so in the way that the machine gets made. So, all right,
I have one more question.
what's up? What you got?
So, going back to, I'm blanking on it, the genetic modification. Would that be considered a violation of God's work since we're manipulating what He's created to create new things, or is that just another example of?
You know, I don't know. I don't know. I don't think you can make a clear case either way. You know, you have to remember that at the time that we're presented that we were created in the image of God, it's in the context of God as creator, right? And so part of that, and I think you can make a really good argument that part of having the image of God is to be creative and to be able to actually create. Now we can't create out of nothing because we're not God, but we can create as well. And then so is that just exercising the image of God in us, or is that a violation of God's design? I don't know. I think the best way to answer that question is, are you making something that is obviously a distortion of God's design? If the answer to that question is yes, then I think you're doing something that you probably ought not to do. If you're making something that I think is just a, like taking something to its fullest extent of what's possible, then I don't know if you're necessarily doing something that you ought not.
What do you mean, like a distortion of God's design? 'Cause I mean, like we use trees to build houses. That's not really what
Yeah, I
God
don't
designed trees is.
Yeah, I think that's fine, and that's an exercise of our creative abilities of taking.
But how do you know what's
where you draw the line?
yeah, or what even defines
God, music, and surfing.
what even defines like what you said?
Yeah, I think it would be a matter of where is the glory going, you know?
So if we're building a house for our own glory, then it's
Then you have a problem regardless of whether or not you ought to be building a house, right?
So
And
it all comes down to your intentions.
no, not necessarily. There are also things that I think are clearly wrong because you're messing with something that you shouldn't. I'll need to think about this more and do some reading on this issue because I haven't done as much thinking on this idea of genetic engineering as I probably ought to. You know, I think one clear some areas where I have thought about it is, you know, if you can genetically modify a plant that doesn't result in any disadvantage to consuming that plant, but actually makes it to where that plant produces more output and provides more food for a human population, I think that's good.
Okay.
And I think that would be an example of which we are using our creativity to do something that is good, right? But if you're doing something just to kind of see what we can possibly do, I think you're doing something very dangerous at that point, right? If all you're doing is trying to see what you're capable of, that seems like a Tower of Babel situation to me, right? Building a tower wasn't an issue, right? There wasn't anything wrong with building a tower. It was what they were trying to accomplish and who was getting the glory.
Oh, okay.
So I think you have certain times where you're doing something that's clearly an issue, and then other times it becomes an issue because of who's getting the glory.
Okay.
But yeah, so I would say, you know, like making spiders that have, you know, chlorophyll in them, that I don't see any real significant advantage to doing that.
what I heard is most of the time that people are upset with that is when the organism that they like genetically modify not only doesn't benefit from it, but is like messed up because of it. 'Cause
Right.
that's like animal cruelty.
Right, is significantly harmed, right? And then you have some issues as well.
That's the only time I've seen like a clear cut. This is why it's wrong.
Yeah.
I haven't heard about the plant spiders before. What's harming them with having the?
Because spiders aren't meant to have nucleus.
Or,
So it's just like.
or yeah, but like I
And
wasn't
so
sure if
they're
there's like a
messed up.
specific thing that was.
Yeah,
affecting.
you know, I actually,
Like everything affects them.
Okay.
yeah, I've got some thoughts that I can share with you. Bring that back up on Monday, and we'll talk about that because I just thought of an ethical theory that you can use to
actually have something to say about that. So.
Okay.